Dzień dobry Państwu.
Hello, ladies and gentlemen. My name is Katarzyna Mucha, and on behalf of the Molecule Management Board, I would like to welcome you at our quarterly meeting. Now we are meeting after the publication of the yearly annual report for 2023. We'll be talking about the progress in our research, both in clinical, preclinical stages and discovery, and also we'll talk about our business affairs. Today with us we have Marcin Szumowski, Samson Funk, Chief Medical Officer, member of the board, Zbigniew Zasłona, Chief Scientific Officer, and Sławomir Broniarek, Chief Financial Officer, and also the Management Board member. We have planned this meeting for around 9 minutes. You have the original track, so mostly in Polish with some English excerpts and a translation interpretation into English. First, we will hold the presentations and then we will hold a Q&A session. Please send your questions in the chat. The presentation is already available to you on the website in the tab Investors. Thank you for giving me the floor. I would like to welcome you at today's meeting. Today we will be discussing mostly what we published in our annual report and also the latest news about the progress in our programs. First, I would like to summarize what we have managed to achieve in the course of the last few months and also to set out the objectives for the near future. The main overarching objective of the company in terms of our pipeline and our research projects both in the clinical and preclinical stages consists in ensuring the possibility of having final readings of the clinical projects that we are holding. As you might know, recently we have been recruiting actively patients for our clinical trials, both for OATD01, where we already have FDA and MHRA approval, so for the United States and the United Kingdom. And we are also waiting for approval for the EU member states. In terms of OATD02, we already received the approval of the Commission to increase the dosage. The results are very optimistic, and Samson will be telling us more about it. So we have an increase. We have been observing an increase of our markers or arginine. So at the stage of the 10 milligram dosage, we have not observed any toxicity. This opens us the window to increase the dosage yet further. We are also very active in the area of implementing various AI tools in drug discovery. Our aim is to confirm the activity and drug properties of molecules which are first pre-selected by AI models and also our own customized model which we will be teaching, which will learn from us. Here we are involving both our internal team and also we have started external cooperation with foreign partners. As you might remember, in July 2023, we had an SPO which gave us 50 million Polish zloty. At the end of last year, we have demonstrated the first effectiveness of molecules in the lab targeting mRNA by way of binding of the right particles of RNA. So we have managed to block the translation of undesired proteins. We also have progress in protein programs. so mostly two programs regarding the ubiquitinase. Today's presentation will focus mostly on clinical programs. Samson will tell us more about it. And that is where we build the greatest value and where we are observing the greatest progress, and where the final readings may significantly change the way that the company is viewed and valued in this year or in 2 years. In terms of the pipeline, there are no new things, no new elements, but we can see progress. The largest progress is in the area of the molecules targeting mRNA. We are very proud and satisfied with the fact that the patients also in OATD01 are already receiving our drug, are being administered with our drug, and I hope that we are anticipating a snowball effect. This first patient is the biggest hurdle that we have to manage, and as we progress, as new sites open, then we have this avalanche effect, the snowball effect, and this is, and we hope that our end of year objectives with regard to the number of enrolled patients will be met. Now I will give the floor to Samson.
Okay. Good afternoon. I'm telling you now something about the OATD01 study, which is our double-blinded randomized study in pulmonary sarcoidosis. We're talking about 3 geographic areas where the study will take place. First, the US. We have received FDA approval, as you know, and we are currently initiating and activating the sites. For the moment, we have 4 sites activated, and they are already screening patients. These patients basically come from their patient pool that they already have, and they are already in the sites. Once that patient pool is exhausted, then they will be reliant on new patients that come. For this, we are starting quite a lot of campaigns, and I will discuss this in a later slide, to create awareness of physicians outside of the sites and patients particularly to be willing to join the study. But I come back to that in a moment. The second geographic area is UK. We have received the MHRA approval end of last year, and we have the first patient already enrolled. That first patient, actually it's a female patient, is now in week 4, and so far no adverse events. So the patient has not complained, which is a very good signal. Although this is a double-blinded study, so we don't know whether the patient obviously has OATD01 or placebo. Nevertheless, the patient is doing fine. The other 2 sites in the UK, we have 3 sites in the UK currently. The other 2 sites are coming on board within the next weeks. Geographical area number 3 is the EU. As you are aware, we have resubmitted in February to the EU, this time without Poland, but within the same countries that had already approved. So far, we are in the plan. The countries have not raised any new questions. So we anticipate that they will basically approve at the latest time point possible. That's what is the normal regulatory standard. So they always tend to wait up until the clock is out. So that is going to happen in June. This slide on the right depicts the geographical spread of our study. Again, to remind you, each patient is treated for around 12 weeks. We will have around 100 male and female patients all with active pulmonary sarcoidosis, which is a relative rare disease. And we will have around 24, 26 outpatient sites in the EU and the US. In the US, negotiations are somewhat more tricky than in Europe, where in Europe, as you know, that we have governmental healthcare systems. In the US, it's a free market, so negotiations take a little longer because the sites want more money, we want to pay less money, and of course, we are very keen on having not too much overspent because that we do not want to waste your money on unnecessary things. So we are negotiating very hard, and that takes sometimes a little longer. However, we do think that we will still get the best value. Again, the aim of the disease is to stop the disease progression and to prevent organ damage to happen. And this is done via a very innovative tool, which is called PET-CT. There is one at the start of the study and one after 12 weeks of treatment. And this is our major endpoint. We want to demonstrate that the granuloma, which is the hallmark of the sarcoidosis disease, can be either halted or even reduced. Of course, we will also try to improve the clinics to boost the lung function, to prevent lung dysfunction, to have a better standing of the lung, i.e. certain criteria like forced vital capacities should improve. And very important also, the quality of life should improve in those patients, something that all regulatory authorities nowadays and the societies are looking for, which is a basic requirement. Okay. Coming back to the US, this is not only valid for the US, but also for UK, and it will then also take place in the EU. Patients are usually in the sites, but not all patients are in the respective specialist sites. So the first thing they do is, what the sites will do is to check their patients in their sites. Once these patients, or once this patient pool is exhausted, then they will rely on new patients. We do that too, but we will we have a lot of campaigns with clinicians, i.e. with GPs, with doctors outside of the clinical sites, but also with patient organizations in every country to make patients, but also doctors aware that there's a study ongoing that may be of interest to them. In the US, for example, we have already, our medical lead has already gotten some phone calls from US patients who were asking about sites in the US who would be open to get enrolled. So you can see that the first fruits of these activities are already coming. So we will visit conferences and make our study well known within the community. With that, we hope that we get a very quick take-up of our studies within the combination of being known to the medical community, but also even more important, being known into the potential patient community. Okay. I'm switching now to the OATD-02. This is our first in-human phase 1 oncology study with the first-in-class dual arginase inhibitor. We are testing this compound in solid tumors. This is not a double-blinded study. This is an open-label, single-arm study with a dose escalation. We need to explore what is the highest dose possible, and what is, of course, also the dose that may have a clinical effect. This is not always the same. But in general, the higher you can get, the better for you, because the higher the chances of having a positive effect. The study is taking place in Poland, in 3 sites, as you know, in Warsaw, in Otwock and Bydgoszcz. And it's a tablet. It's taken once daily as a monotherapy. Remember, in phase 1 studies, all patients must have had standard treatment, and there's no other medication available anymore. Most of our patients that we have already had 3, 4, 5 lines of therapy. Some of them come in with 5 lines or 6 lines of therapy and are basically, they have no other chance than to try OATD-02. So it's a very, very sick patient population that consists of colorectal cancer, colon cancer, ovarian cancer, pancreas cancer, a very nasty tumor, and renal cell cancer. And you can imagine these patients are, as they are very sick, not all the patients can stand in the study up until the end of the studies. Some already progress whilst they are being checked by the clinician. We're trying to mitigate this, but this can happen. You will see this happened once in our study. And some of those patients progress during our study, which is an indicator that our drug may not yet be so effective. We have foreseen 6 doses. We are currently in dose number 4. So the first patient for that 20 milligram cohort is currently being screened. I signed the allowance form last week, so he's now in screening. At the last meeting, I think we talked about the 10 milligram cohort. Now we are in 20, which means the 10 milligram cohort was released by our safety review committee as safe. That's a very good signal. I think even more important or more interesting is that our biomarkers that we measure in our patients were somewhat as expected. Both of our safety markers, orotic acid and ammonia level, were completely normal, so no issues there. And our efficacy marker, which is the arginine level, was very stable. We could verify that there is a trough level, and of course, it fluctuates between one and the other patient, but within the same patient, we have a very stable level. So we could, if you want to have a firm statement, we could say that we can see a certain a drug level now being as we anticipate. Obviously, the exposure of the patients is still on the lower end. So we did not expect any major efficacy signals, which is obvious. But with the doubling of the dose from 10 to 20 milligrams, we do expect that we will eventually get into a range where we can see some efficacy results. Definitely, our efficacy markers have already responded, but again, the exposure is still a little low. Next one. This is a so-called swimmer plot. I think that it has been shown already. Every time we will show this swimmer plot to you, there will be some changes because it's a living document. This is a very transparent document that shows you about every patient in the study, what tumor type the patient has. You see that on the left. see colorectal cancer, renal, colorectal, pancreatic. You see the dose cohorts, 2.5, 5, 10 milligrams, and you will see how the patients developed over time. Let me focus your attention to the one on the top, the 2.5. This was a colorectal cancer patient that was on study for the entire length, 24 weeks. There were 2 checkpoints. At those checkpoints, which is every 6 to 12 weeks, according to international standards, the patients get a CT scan, and you can see this blue shade, that this is that the patient was stable, i.e., the tumor did not significantly grow during this period of time. Then the patient was entered into the rollover study, which is the red bar, but after a while, 38 weeks, the patient finally progressed, which is not unexpected, but it was unexpected that the patient would stay in study for so long. But the patient is still alive. You can see this in the green bar. The last patient of this, for example, is the colorectal cancer, a female colorectal cancer patient. This patient was the last patient in the 10 milligram cohort, and you can see the patient is still ongoing in the study and will likely have the first checkpoint in 2 or 3 weeks. And every time we will present, there will be some changes as we go along. The patients, obviously, the bars will get longer, and then you can see on how the patients develop. I think that was my update, and I'm handing over to Zbyszek for some more activities on the non-clinical area.
Jako przedklinika, jako program
first class preclinical program, we try to support the programs from a scientific side to increase their value. The more we know about the mechanism of action of those molecules, the better the doctors and the patients feel. As far as OATD01 and chitinases, what we have been working on for some time now is showing the mechanism of action. Chitinase, chite is chitinase. This comes from chitinase, chitinase, and currently we have proof coming from our research that KITE1 was down-regulated, and it showed that the substrates are sugars that change the post-translational coding of protein, and the glycolization process is changed in such diseases as sarcoidosis, where chitinases contribute to this disease, contribute to the inflammation and fibrosis, and our OATD01 is able to inhibit this state. We have made a publication, one of the 3 publications on chitinases that will appear this year in scientific reviews. The first one is going to show the context in which we want to develop the understanding of chitinases, and the second is going to be more mechanistic. As far as the support to OATD02 program, we have stressed that we are the first-in-class drug, and we have stressed the metabolic changes. We are also working on a publication in cooperation with the University of Mannheim, and precisely with the team responsible for mass spectrometry imaging, which has visualized our hypothesis that the metabolic activity of cells changes under the influence of OATD02. We can see on one side that arginine, the amino acid, which is indispensable for the healthy functioning of the immune system, is practically non-existent in cancer environment. Whereas after adding our drug, it increases. The proline and spermidine are polyamines that are inhibited. The polyamine synthesis is something that makes tumors, cancer grow. So this is something that demonstrates our hypothesis that we have the improved immunoactivity and some improved immunoactivity through the increase of arginine and also the inhibition of polyamine synthesis. As I have mentioned, because these programs are very active in our company, we have engaged many specialists. Because these are first-in-class targets, a lot of effort was focused on the publications I've mentioned and on looking for the mechanism of action proof. USP7 is one of the more advanced programs which we have supported even more as far as medical chemistry is concerned. This is we want to obtain a lead molecule. We want to have optimal drug-like properties, and although the entire package of data has shown that our compound works quite well, we have decided to work a little more on the preclinic stage in order to save time and money in later stages. So as far as the USP7 program is concerned, this is the program where we are testing, like Marcin mentioned, we're testing the AI solutions. We ask a specific question to our AI tool, what specific properties we want to achieve in a molecule, and the molecule development is now sort of driven by the AI platform. As far as the mRNA platform is concerned, once we obtained the proof of concept, so we designed molecules that would bind to mRNA, and the protein which is coded by the mRNA, the translation of it is inhibited. Once we have seen it, we decided to look for CRO companies and for academic research centers. Now we are ready with the offer. Recently, with Marcin and Maurycy, we visited BIO and we presented our offer, and we will extend our offer of cooperation to numerous partners. We are able to find a region in mRNA that has the potential to drive a program, and we are able to continue in that program. These are some of the results that we have not been able to show. We want to show here how we have achieved the proof of concept in different molecules. We found a structure that has high affinity to small molecule compounds. We've been able to verify the structure in the shape. Then we have confirmed functionality using the omics technique. We also need a specific sort of tool set and techniques that will allow us to select compounds. So we have created a virtual library that has some 500,000 different compounds that to a certain extent have high mRNA affinity. We have conducted virtual screening using this library, and the best hits, the best molecules binding mRNA were selected and then confirmed with the SPR method of a CRO company. This allowed us to select 30 hits which had multiple bindings, and 5 hits had the binding curve less than 10 nanometers. So the best compounds we've selected with the best drug-like properties were then exposed to cells. The cells included our target protein, and which we previously confirmed in terms of its functionality, in terms of its turnover. So, as you can see in this Western blot test, we have the housekeeping gene in the bottom row that shows the health of the cells, and in the upper row we have the changing band. We have our target and the level of inhibition of this protein and the level of translation correlated with the results shown with the SPR method. In the meantime, since we obtained these results, we have been able to repeat these results, to confirm them, verify them with the SHAPE method, and we have observed statistically significant changes. So we've been able to add more pieces of information that supports our approach.
Thank you, Zbyszek. And now we will focus on the numbers. And I will show you how our efforts in our projects have translated into actual money. Let's start with the profit and loss account. Looking year on year, we can see that not much has changed, although if we have a closer look, we will see that the inflows are lower than last year. It's because of the end of the grants for the previous perspective until 2023, but you can see that there is a significant dynamics. What happened? At the cost of administrative costs, I would like to draw your attention to the fact that we try to keep reasonable conditions, so we have spent less. However, the early stage programs have dominated here the expenditure of our company. This is spent on programs mentioned by Zbyszek. But when we look at our revenue, we can see a dynamic increase for the year 2023. It's because of the return of the tax, which we managed to obtain from the tax revenue office. This is related to the Galapagos program. So as a result, we have a comparison of these numbers, as you can see on the slide. The main driver of the costs were the early stage programs. When it comes to our balance sheet and our assets and liabilities, here we can definitely see an increase of the balance amount, which results from the increase of the clinical programs that we are developing. You can see the amount of over 40 million. I will talk about it in a moment. When it comes to the liabilities, we have the increase of the capital. Thanks to your input, we are able to close this matter very well. As already mentioned, I will try to compare the 2 years together and show you that what happened on the level of clinical trials and preclinical trials translates very directly onto specific numbers. As you can see, the expenses on R&D, that was over 40 million in 2023. That's 40 additionally with 10 million for the early stage programs. So when we sum it up, you can see that the dynamics on this expenditure is huge. While at the same time, we have managed to keep the administrative expenditure at the same level or even lower. So we have an increase of the 30% over year on year of the dynamics, but this is the result of, again, the result of dynamic progress in our clinical programs. Compared to last year, it's an increase 2.5-fold. At the end of March 2024, the cash position is 54 million Polish zloty. We have 114 employees, and I don't have the exact number, how many people with PhDs we have, but we definitely have more than 50 hold a PhD degree. This slide presents our situation with regard to the grant applications that we submitted. And here, despite of a lot of data, let's focus on some of them. Let's see at the penultimate position here. We have applied for a grant for an AI tool to pre-select molecules that would bind with mRNA or with proteins. This is what Zbyszek mentioned. This grant was submitted to, as part of a consortium. The qualified cost is 42 million and the funding request is 32. Summing up, one program, we've got one program, the mRNA program, which already has a grant agreement, and you can see the clocks here, the pictograms, which tells us that the clock is ticking, time is running out. So it means that time is running out on the institutions that are to make a decision. Some of the programs have been waiting for a decision for a long time. Some of the applications were submitted in summer last year, and the decisions will be made when we see PARP. The decisions will be taken by PARP in the third quarter of this year, so that's late. We hope that NCBR, the institution NCBR, will be more swift in its decisions. We are also hoping for a quick decision with regard to the application submitted to NIH. I think that by June we should have a decision about whether we will get the financing. We would like to present the slide to you to present our plans for this year and next year. You can see that we will put emphasis on clinical programs where the expenses will be around 60 million Polish zloty. When it comes to expenses on early programs, it's around 37 million Polish zloty. mRNA will probably involve expenditure of 25 million Polish zloty. The rest is general administrative. That's around 28 million for those 2 years, 2024-2025. We hope that the decisions that I presented to you will be positive and that in the second or third quarter, we'll have the decisions that will total in at least 75 million Polish zloty. What we are also anticipating are, as a source of financing is the partnering transaction, so the public market and also public market. So our focus is on the partnering transaction, and I hope that this will allow to cover the needs that I mentioned. But more about this deal will be explained by Marcin in a moment. Thank you. So a few words about business development of our company. I would like to focus on the last conference, BioEurope, that we participated in and presented numerous of our programs. They enjoyed the interest of a lot of partners, potential partners. This conference was a very good signal. Generally it indicates that there is an increase in activity. This conference resulted in a record number of meetings that we held. As you can easily see, the main interest that is on the rise is regarding OATD01. That was the topic of the largest number of meetings, 20 meetings, and we also had returning companies coming back for meetings, including the big pharmas, the main 10 companies from various parts of the world. And also what Zbyszek mentioned, huge efforts put into describing the mechanism of action, the use of genetic data with regard to transcriptomics, and a number of technologies where the therapeutic objective, which was unknown to most researchers, only some of them knew chitinases, and only some of them were expressed in pulmonology, which use this biomarker, specializing in sarcoidosis and also other institutionary lung diseases. This objective was unknown to them, so it is always more difficult to convince someone to spend a lot of money on an objective which is not validated in clinical research, because it is still unknown whether it works. So we will continue to publish further articles in various scientific journals, which have a huge impact factor. We'll get the academia, the people acquainted with the topic, and we will present the important role of chitinases. And we are also getting ready for a joint publication, which, using various algorithms and genetic data and AI methods, shows that there's not just a correlation, but there's also causality. So the overexpression of KITE1 is the reason for the disease and not its result. This is what we're going to demonstrate. This data helps us convince people to believe that we might be talking about a breakthrough therapy which addresses the origins of the disease and can cure the patient. So a lot happened. Also, there was a lot of going on also in other areas, also regarding OATD02, mRNA as well. And this is something what we are also anticipating. At least a partial signal for activity, so a partial response, because this is a trigger that will also help interest companies in getting into advanced partnering negotiations with us. And mRNA, which we are currently exploring, we can see significant interest in our work. A lot of pharmaceutical companies are searching for methodologies which would be the most effective, which would be adequate to what they want to achieve by adopting an alternative treatment of various diseases. So here we are trying to customize our offer. We try to collect as much data as possible for various therapeutic objectives to allow a long-term cooperation or to be able to provide services to companies who have a specific target that they need validated to see whether it is possible to address a specific problem with a specific molecule, and all that with specific money. In this regard, we are counting on more meetings. A lot of them are follow-up meetings. When it comes to the category you can see other, they include meetings with companies, with pharmaceutical firms, where we can strike cooperation, or with investment funds which are interested in the company as an investment target. Here you can see some notes from the meetings that we are continuing to hold, where we are going more in depth with our negotiations, with our talks. What we are observing is increasing interest from big pharma companies. We had several very promising meetings, and we can see that the appetite for risk, the tolerance for risk is increasing, especially in Asia, where we continue to hear that they will be happy to buy licenses once we manage to demonstrate more. But there are a lot of companies which are intrigued by the mechanism of action in various indications, and the talks are ongoing. All in all, a very good conference. We are counting on similar results on the next conference, BioStan, that we are participating in 2 months.
Now let me offer a summary before we move on to Q&A. The element that we want to achieve at the end of the year is the interim data analysis by an independent, unblinded committee after completion of dosing of approximately 50 patients. This may seem like a far-reached target because we have one patient now. There is a non-linear progress of in enrollment. We are talking with multiple centers, so we think we are going to speed up very soon. We are also starting a number of other activities in cooperation with the Foundation on Research in Sarcoidosis in the United States, because we want to promote and increase the number of patients in the US as soon as possible. We plan to finish the study at the end of next year in 2025, and we will then have an answer, a clear answer, whether we see a result of our drug versus placebo. When talking with different companies during business meetings, we can see that all our potential partners are very intrigued by our endpoints. So the PET-CT imaging results, this is a new endpoint. The key opinion leaders and the majority of doctors agree that this is the most objective form of confirming the effect of our drug in a patient's body, and that it has the best it gives the best prognosis for quantification and the proof of concept. So unfortunately, we are very unhappy that we haven't been able to implement the study in Poland, but we're not giving up. We're still trying to to reach the Ministry of Health with through the influence of the scientific community, with the message that we really wish to dose the drug, to be able to give the drug to Polish patients as soon as possible. As far as OETD02 is concerned, as stated by Samson, we can see a significant increase and stabilization of the PD marker, so the level of arginine in patients dosed with 10 milligrams. We are waiting for the results for the 20 milligram cohort, and if there are no if there's no significant toxicity in 20 milligrams, then we can see that the therapeutic window will expand, and we hope to receive the first efficiency signals and being able to establish the maximum tolerated dose. The escalation, the dose escalation process is difficult to predict, so we don't want to make any forecasts as to that. We may reach the maximum tolerated dose quite soon, quite in the near future, or maybe the dose will reach much higher values, and then so we'll have the higher safety margin, but then it may take longer than the beginning of 2025. It may be more mid 2025 or the end of this year, of the next year. As far as the AI platform is concerned, we have been investing in the development of algorithms. We have used a number of licenses, and the molecules generated by AI models are already being tested in our laboratories. We would like to be able, based on the data from all our programs, to be able to teach the AI to. Of course, as we know, AI is as good as it is trained to be. So we want to be able to speed up the discovery of molecules that are already optimized thanks to the learning, AI learning. So we'll be able to soon to obtain molecules that would have much less sort of deficits in their design, because even small challenges delay our studies, as we can see with the first molecules, which are very good. But if had we been able to optimize them from the beginning, then we would have needed less clinical research, less time, and we've been able to achieve the clinical phase much sooner, and we would not need so many additional testing. So we have now employed an active team with developing the AI tool and feeding the tool with data, and hopefully developing drugs faster. As I mentioned, the bio conference, from the business perspective, the partnering agreements are the most important objectives, and the biggest interest is generated by OATD01, but also 02 and USP21. The USP7 needs still to achieve a flawless molecule so that we can promote the program in business terms. So we are now trying to use both our contact networks and our supervisory board, our key opinion leaders that have broad business relations in order to be able to find in the months to come, to find a company that will believe in changing the situation of patients with our molecule. And this will be a very nice moment for us. So thank you for your attention, and now we'll be happy to answer any questions that you may have. Thank you for your presentation, and now we move on to questions from the audience.
The first question, did the January transaction that happened in January this year with regard to mRNA between Roche and Rx Therapeutics is a good benchmark for the company's actions and the possible commercialization? Well, we hope that indeed it is. We try to benchmark by using all data and all historic transactions. So we have a model that we have built that calculates the statistical likelihood, the mean averages. This transaction fits into this area, including some error margin. So it is a good reference point, a good benchmark for what is happening on the market. And the next question, Business Radar in its latest publication indicates that as a result of the meetings it held, the company has concluded 11 CDAs, so confidentiality agreements. Can you corroborate this? Well, if it's the data that we indicated, then definitely. However, we try not to announce details, ongoing data, because this situation is very dynamic. So definitely, we did sign this number of CDAs. Historically, I would prefer not to indicate how many of them are active. In materials of brokers, after one of the conferences of the company, there was a comment regarding about caution regarding OATD02. Can you tell us more about that risk, and what is the likelihood of this risk coming to becoming an actual problem?
I don't actually know what risk was mentioned. Oh, uric acid is one of our safety markers because of the disruption of the enzyme cycle. Obviously, when you interrupt the cycle, it's like in a traffic jam. When you interrupt the cycle, you get you can get some congestion prior to the blockage, and it's one of our safety markers that we regularly measure. If uric acid would eventually get too high, this could be one of the limiting factors of the dose, which means if we increase the dose further and further, at some point in time, it can be that the uric acid reaches a level where it gets toxic. For the moment, at 10 milligrams, we have seen virtually nothing. It was completely normal. We will see with 20 milligrams. That's the double dose on how this will develop. But for the moment, we do not see any major risk here. By the way, we have not seen any major adverse events. And as an oncologist, I do expect that an active agent will eventually also have effects that you may not want, which is a side effect or toxicity. So far, we have seen a little more from the numbers, a little more of adverse events in 10 milligrams than in 5 milligrams. Which is expected because you double the dose already. But the severity has not increased. So the highest grade of severity was grade 2. And as you may be aware, a grade 4 would be a so-called, eventually a DLT. So there is a margin, a clinical margin that is still significant. And from the non-clinical data, if I'm citing correctly, my colleagues correctly, we still have a margin of tenfold to the toxic dose with respect to the non-clinical toxicology.
So maybe just to clarify, I want to speak in English, so Samson can correct me and he gets what I'm saying. But orotic acid in its own is not toxic. Let's make that clear. So that this is an increase that we observe, that when it's prolonged for a long time, at some point, a very high increase, it may lead to ammonia accumulation, which then is an issue. So that's not a proper, maybe, way of describing that orotic acid and conclusion made.
It's our second safety marker, ammonia level, and that ammonia level was basically completely normal.
Thank you very much. Teraz następne pytanie.
Moving on to the next question.
Seria pytań dotyczących badania OATD-01. Czy spółki nie martwi wolne tempo zgłaszania się pacjentów w badaniu leku OATD-01, zwłaszcza w Stanach Zjednoczonych, i zważywszy na brak na tę chwilę zgody w Europie?
And the lack of consent for the trial in the EU. How important is the US in this trial? Can you compare this reputation to other trials and reputation this trial to other rare diseases?
To the latter part of your question first, no, we cannot. If you look at all the other agents that are currently in development for pulmonary sarcoidosis that also do studies in the US, and it's a competitive situation, so there are a lot of sites where there are 3 or 4 studies in pulmonary sarcoidosis ongoing, but they are targeting completely different population, which is for them an advantage, for us not necessarily, because when you have the disease, at some point in time you will get diagnosed by the doctor. Then the doctor will not necessarily immediately send you to a specialist because the disease is still in the beginning. This is the population we are targeting. So we basically are targeting those very early patients that are not yet that sick. Eventually, those patients will get corticosteroids over time. Many of them go into spontaneous remission, but many of them will also need corticosteroids. And we would be capturing those before they get corticosteroids. So the target is that eventually you would never get corticosteroids. As you know, corticosteroids is very effective, but it comes with significant, sometimes deleterious side effects. So we could eventually prevent these. So this is a very early population. The other compounds, Xantria and all the others, they are targeting the patient populations that are already sick. If you look at their inclusion criteria, these patients already have pulmonary damage. They may eventually also have 2 or 3 other drugs already ongoing, like corticosteroids plus an immunosuppressant. So they are heavily bombarded with medication, and their medication comes on top. So it's adding toxicities. They will try to reduce the corticosteroids in those patients. So that's the typical patient profile of the other studies. So we are targeting a different study population. But this is also our challenge. those patients that are sicker, they usually get to this specialists sites. We do also have patients in those specialist sites. Nevertheless, many patients that we would like to have are not yet that sick, so they may not be known to the specialist yet. That's why we are doing so many media campaigns, cooperations with patient associations, because if you are diagnosed as a patient with a disease like sarcoidosis, the first thing a patient does is he goes to the internet and checks whether there are associations, whether there is studies ongoing, clinicaltrials.gov that they can join, and then they will discover us. They will discover, oh, there is a study, this is the only study that is applicable for them because they are not that sick yet. They do not have a pulmonary damage and they do not really want to have pulmonary damage. So then they would be eligible for our study. The challenge is that these patients know that we are there, molecules there. And that's the aim of our, let's say, branding campaign, to make this known to the patients, but also to their GPs when they first see the patient and say, oh, you have a sarcoidosis. Oh, I think there is a study ongoing from a Polish company called Molecure. So maybe you go to the internet and check which site is eventually eligible. And we have a website. This is in the slides. You see this girl with the kite. Our study is named Kite, and there's a website live in the US and in the UK currently, where patients can inform themselves and can tick on which sites they are eventually available for them to be taken into the study.
All right, just to sum up this element, this US element versus Europe, surely the trial in the US is important because, firstly, because of the market, secondly, because of a different patient population. For example, the Afro-American patients that were in which the sarcoidosis has more prevalence. So it is important to have this group of patients included in a study. However, the US is much more expensive than Europe in terms of study costs. So we really hope for this NIH grant to cover some of the US expenses. We are trying to maximize the number of centers in Europe and in the UK because there is a huge difference in finance, and we want to manage our money wisely. The question whether we are worried by the slow start of the study, well, the most worrying element was the lack of possibility to give the drug to the Polish patients, but compared to several other rare inflammatory diseases, the slow start is a standard. It is normal that it is difficult to start, and then there is the snowball effect. So we hope to see this effect even before the summer, within the coming months, to see the patients arrive at the centers. Of course, we'd like to speed that up, but it's outside our control, really.
Thank you, Martin. Let me add to that we will have similar campaigns in Europe, but we cannot start this as long as we have no approval in Europe. So this will start then once we have the approval. And one should not mix a phase 2 study with a phase 1 study. In a phase 1 study, we are bound to certain rules of how we can take patients into a study. For example, we can always take only one patient in the dose cohort. Then we have to wait for at least 2 weeks and to see on whether the patient has a toxicity from the drug before we can put in the next patient. In a phase 2 study, like our pulmonary sarcoidosis study, theoretically, and I emphasize the word theoretically, all patients could be enrolled into the study in one day. Of course, this is not going to happen because of the resources of the hospital and because not all patients will come in one day. But theoretically, once, as Marcin said, once we have a few patients, then word will spread. And then we are very confident that we will have a wave of patients that will help us to meet our targets. It's the same as when we had the O2 study. Initially, no one wanted to be the first. And then one of the sites enrolled a patient, and then all of a sudden, all the other oncologists, the other sites realized, oh, the study is starting, and we also want to be on board. The train is leaving the station, we want to be on the train, and now we do not have an issue with patients that are eligible for the study. It's just a matter of whether the patients are eligible for the study. So this will be a similar effect here.
Thank you very much. Thank you for your thorough response. We proceed with further questions.
What, according in the opinion of the management board, is the next milestone that can boost the valuation of the OATD-01 program? The nearest milestone or the one that can boost the development, because these may not be the same. The nearest milestone is obtaining approval in Europe and starting the more centers in the US, starting centers in the US, so increasing the enrollment of patients. This is the indicator that we are able to follow, and that will be decisive. But the valuation of the company can also be increased by a signed partnering agreement. The larger the better. So we also hope for a cooperation in mRNA platform and maybe in one of our earlier programs. So these may be small milestones and then a large milestone we hope for. And of course the interim data, because this will either be positive or negative. With positive information, we should have a big change, because it would also make the big partnering agreement more likely. If everybody's listening closely to our presentation, we hope this can also make a change.
Can you please explain what interim analysis is about? Is it possible that it will give you information about efficacy to present them to your potential partner? I think we've already covered part of this,
yes,
but just to make it clear.
Let me explain this again. This study is a double-blinded study. And it was very important when we discussed with all the authorities that the blind is not broken. You have to imagine that half of the patients will get placebo, which is a sugar pill, so basically. The others will get OATD-01. So if at some time point the data are released and these patients may fare better that get OATD-01, you will get no other patient to consent to get placebo. So then the study is destroyed. That's why it's so important to keep the blinding. However, the interim analysis is a time point after 50 patients to discuss on whether, number one, the drug is safe, placebo versus our drug, i.e. our drug is safe, and number two, whether we are on the right track with the study. So there are three scenarios on which the IDMC, which is an independent committee, including a statistician, will have to decide. Scenario number one, our study drug is too toxic, i.e. it's not safe, because many of those patients have toxicities. Then they may recommend to stop the study, which is not a result we would like to see. Number two, they would decide, the study is according to plan because there is a statistical claim that we have. Our claim is that we are better than placebo, but not only numerically, but also statistically, which is the very strong hurdle. If they say you can go ahead and you do not need to change, indirectly, it's a signal that our drug does what it's supposed to do. If they say you can go ahead, but you need more patients, that indicates that we are on the right track, i.e. our drug is better than placebo, it works, but we need a little more patients to meet that statistical hurdle. Statistical hurdle means placebo has an effect of, let's say, 5%, we have an effect of 25%, which 25% less inflammation in the lung. So that's 20% better. Or 5 times better. That would eventually, that works with the number of patients we anticipate. If not, but if we are on the right track, we may need a few more patients. But this is also a positive signal. So scenarios 2 and 3 are positive signals. Scenario 1 obviously would not be a positive signal. So that's the three options the IDMC will decide on in the interim analysis. But it's clear that in question of 2 and 3, this would attract potential partners significantly.
Thank you, Samson.
The next question regards the trial on OATD-01. When is the latest that the last patient can be enrolled for this trial to allow the company to make it in time to provide interim results by the first quarter of 2025?
Okay. So it's trochę skomplikowana.
Basically, it's basically the end of 24. So the 50th patient would have to be enrolled in and the 31st December of 2024, because every patient should have 12 weeks treatment. Whether whatever he gets, it doesn't matter. Every patient will be in the study for 12 weeks, which is 3 months. So if you want to have the interim results by end of first quarter 25, then the last patient of this part would have to be enrolled on the 31st of December 24. 3 months earlier.
Thank you.
And now a question regarding finances. A question regarding dotations. What applications for grants have you already submitted, and which ones are you planning to submit this year? I think it will be more useful to come back to the slide presenting the applications. So once again, we have submitted 6 new applications. They are still waiting for a decision. So OATD-01. We also have one for an undisclosed target. We also have one for the AI tool. You might remember that one application was rejected, and we will be using another. We will be reapplying with the next perspective for USP7. And the question is whether we are planning on submitting new applications. Well, this will result from our internal efforts. If our early stages efforts will suggest something promising, we might decide to submit new applications. And as we've already communicated with regard to expansion for OATD-02, we are in early talks on cooperation with very recognizable, very well-known clinical centers. And then we will be considering applications. If translation studies suggest this is the right way to go, we will probably also be submitting an application in cooperation with a clinical facility partner. However, that might take some time. But yes, we might submit such an application. One more question regarding grants. Two questions actually, but I will combine them. With regard to the mRNA grant, did the company already receive the first advance payments? And then have some advance payments already been received by the company? And are you expecting some advance payments in the second quarter? It's a good question. I would like to know the answer myself. With regard to mRNA, this is not that easy. We've already submitted the first motion for payment, so we are waiting for the decision on PARP's decision. Whether we've received funds? Well, definitely we've received some funds as part of the donations. We will have the report very shortly at the end of this quarter. For the end of this quarter in April. So you will get the data definitely published in this report. Another question regards business development. Are you maintaining that you're going to sign at least one large partnering agreement by the end of 2025? Yes, we uphold this plan. At first I thought that the question regarded 2024, but no, by 2025, definitely.
A question concerning OATD02. Based on the data from patients in phase 1, OATD02, do you see a correlation between the inhibition of the arginase and stabilization of the disease?
Currently, not yet. As I said in my presentation, the arginine level has reacted as we would have liked, as we expected, but it's still on the lower end of the spectrum where we expect efficacy. So, and please do not forget that the last patient, the two last patients, they have just been on study for 6 weeks or 4 weeks. That is still very, very early. There's a good reason why the interim checkpoints for tumor response are taken somewhat between 6 and 12 weeks. That's usually the first checkpoint, via a CT scan. And the reason is that the tumor will also have to react. If the tumor reacts to our compound, then it will take time to react. If the tumor does not react, it will also take time. So 6 weeks is a little short to make any judgment. For the moment, we do not see a tumor response. Whether it's a stabilization, we will see in a few weeks.
Quite a similar question, but this time about the arginine, the biomarker. To what extent the 10 mg dose changed the arginine level in reference to your expected level? So you expected, for example, an X% of growth of arginine, and how far are you from the expected target?
This is difficult to say. We have only data from 3 patients. So, of course, all the patients are above the level of, or in the level of where they have where there is some therapeutic efficiency to be expected. Which level we need to achieve in order to get a tumor shrinkage or a tumor response, that can currently not be finally determined. This may also depend on the tumor, because to give you the example, colorectal cancer may respond a little quicker and a little stronger than pancreatic cancer. As we know, pancreatic cancer is one of the most difficult tumors to treat overall. So, and the reason why is for the anatomy of the pancreatic cancer. So, depending on what tumor we have in which patient, eventually we may get an earlier result, but eventually we may get a later result. That can currently not be finally determined.
Dziękuję bardzo.
In patients.
Thank you very much. And one more question about OATD02. In your presentation, we've seen about the non-evaluable DLT in one OATD2 patient having the 10 milligram dose. Can you tell us more about it?
There was one patient who was enrolled with the colorectal cancer. At the time of the enrollment, the patient was relatively well, but he had quite a few metastases. So there was a big metastasis in the liver, lymph nodes, and lung. So quite a few metastases. So during then the patient was still eligible for the study. The patient was enrolled and received the study drug. Then after around 3 weeks, the patient had to return to the hospital because there was a fistula developing in that patient. So the patient had a discharge. So the patient was then hospitalized and a CT scan done. And it was then found that the patient has progressed in the disease. And the disease progression is when the disease progresses, we have to take the patient off the study. Unfortunately, this happened before the DLT phase. The first 4 weeks were over. Obviously, this was not study drug related, so independent of the study, the tumor has progressed. So the patient was taken off study and then treated in the hospital. For the progression, for this fistula, for the progression of the disease. Later on, so the patient was operated, and patient died then 3 weeks later after the operation. But at that time, the patient was already off study for 4 weeks. So there was no connection to the study drug. But since the patient progressed within the DLT phase, the first 4 weeks, we had to replace that patient and bring in a new patient.
Thank you very much. One more question. It might be the last one, actually, as I look at the time that we've scheduled. A question regarding OATD02. In inhibition of arginases, there is the accumulation effect. So the change after 12 weeks, are they significant then at the onset?
I didn't really catch that question. What was the? Oh, did you?
I'm not sure.
Whether there's any accumulation.
Where is the 12-week coming from?
Ale czego
akumulacja? We didn't see any accumulation in the arginine level in any patient. I mean, the best data we have is from the 10 milligram patients, but they are all stable.
Może się trochę pomieszało, bo 12 tygodni to jest OATD01, tak? A 12 weeks is not any time point in your study OATD02 study.
No,
right?
It's not.
So
maybe this is mixed up with the OATD01. OATD01, the patients will be treated for 12 weeks with OATD01 or placebo. And the 12 weeks was chosen because all our advisors, the pulmonary experts, told us that 12 weeks is enough to demonstrate the effect of OATD01 on the granuloma in the lungs. If you take it longer, then you can take longer, but you then risk, anyway, it costs more. It may not be better, and you may run the risk of patients dropping off the study or toxicities. So they all said 12 weeks is enough, and that's what we are doing.
A jeśli chodzi o, że było pytanie, czy tam się zablokuje, tak? Że, znaczy to nie ma takiej sytuacji, żebyśmy zahamowali całkowicie arginazę i żeby ona nie działała. To jest właśnie ten moment, kiedy chcemy zahamować ją na tyle, żeby ten wzrost był argininy. My sobie postawiliśmy 3 razy tyle. 3 razy w porównaniu do kontroli. Ten oczekujemy po badaniach sprzed kliniki, że będzie miał odpowiedni efekt biologiczny. Natomiast nie ma takiej sytuacji, że zahamujemy całkowicie całą arginazę w organizmie, bo to nie byłoby zdrowe.
Być może chodzi o jakiś efekt ponadproporcjonalnej
Maybe is regard the proportional inhibition. We will try to go into the question back. So the idea is not to go overboard. That's why we assumed this threshold.
We have not seen any accumulation of arginine in any of our patients in O2 study up to now. So.
Dobrze, bardzo dziękuję w takim razie.
Thank you so much. We'll try to go back to the question, ask what exactly was meant, and we'll answer it later. And for now, I'd like to thank you for your time. Thank you for the time of the management board members, and we would like to thank for all the interesting questions. The recording from this meeting will be posted on the company's YouTube channel. The presentation is already available on the company's website in the investors tab, and as always we encourage you to contact the investor relations department, and we wish you a very good afternoon. Thank you for bearing with us.